All right, let's get into it. Today, we're basically playing detective. We're on the trail of a cancer that is, for lack of a better word, super smart. It seems to have a counter move for every single attack we throw at it. So, our mission today? To crack its code.
First things first, let's meet the villain of our story, triple-negative breast cancer, or TNBC for short. And believe me, in the world of cancer research, this one is known for being a particularly tough nut to crack. And this isn't some super rare thing either. TNBC actually makes up a pretty big chunk of all breast cancer diagnoses. We're talking somewhere between 15 and 20%. So cracking its code? It's a massive priority for thousands and thousands of people.
So, what does triple negative even mean? Well, think of it like this. Most breast cancers have these little docking stations on their surface, called receptors. Our best drugs are like keys designed to fit into those docking stations to shut the cancer down. But TNBC? It's missing the three most common ones. It's like a fortress with no doors or windows for our best keys to get in. And that's exactly why it's so hard to treat.
Now, our investigation really kicks off with what seemed like a super promising lead. Researchers spotted something happening inside these cancer cells that looked like a total smoking gun. It felt like a clear path to a solution.
So, the first clue is this molecule with a mouthful of a name, LINC01133. You can think of it as a kind of supervisor molecule that tells other genes what to do. And investigators found it was working serious overtime inside TNBC cells, just pushing them to grow, grow, grow. This became our prime suspect.
And as they dug a little deeper, they figured out its MO. This LINC01133 molecule was flipping the master on switch for a major growth engine in the cell, something called the AKT pathway. This pathway is basically the cell's command to grow, divide, and, most importantly, survive.
At this point, the solution seems pretty obvious, doesn't it? I mean, if our suspect is turning on the growth engine, then let's just shut that engine down, block the AKT pathway, and the cancer should just stop in its tracks. Simple.
But, and this is where our case takes a huge twist, it didn't work. When researchers used a drug to block that pathway, the cancer cells barely even blinked. They just kept on growing. The cancer was way smarter than we thought. Our first lead was a total dead end.
This surprising resistance could only mean one thing. This cancer wasn't relying on a single playbook. It had a backup plan, a secret escape route.
You know, the crucial realization was that the cancer had this hidden escape route. The moment its main growth engine got shut down, it just fired up a second one. It had a whole compensatory pathway just waiting to take over, allowing it to keep right on growing.
So the investigators went back to the drawing board, sifting through all the evidence. And they found it. The cancer was firing up a totally different growth engine, the MAPK/ERK pathway. This was its secret weapon, its backup plan to survive that first attack.
Okay, so we know what the backup plan is, but the next big question is how. How does our prime suspect, LINC01133, manage to switch on this second pathway? It couldn't be working alone. It had to have an accomplice.
And here's the secret mechanism they uncovered. It's this sneaky three-step conspiracy. First, our main suspect, LINC01133, boosts the levels of a protein named PRR5. This is our accomplice. Then PRR5 steps in to protect another key protein, a master organizer called IQ-GAP1. And finally, with its protector in place, IQ-GAP1 is totally free to turn on that backup growth pathway. It's a pretty intricate little plot.
So what's the easiest way to think about all this? Just picture PR5 as a bodyguard. Its entire job in this whole scheme is to protect the really important player, IQ-GAP1, and keep it safe from harm so it can carry out its mission.
And this is basically what it looks like. Normally, the cell has a kind of cleanup crew that would find and get rid of IQ-GAP1. But this diagram shows our bodyguard, PRR5, physically getting in the way, shielding IQ-GAP1 from being destroyed. This one simple act of protection is the key to the cancer's entire backup plan. So now we get it. We understand the cancer's entire strategy, a main growth pathway and a backup pathway, all enabled by this clever bodyguard system. And with this knowledge, investigators could finally cook up a plan to actually defeat it, a coordinated attack.
Let's go back to the data, because it really shows why those first attempts were doomed to fail. You can see it clearly here. It didn't matter if they attacked the main pathway or the backup pathway. Neither approach worked on its own.
The cancer was just too resilient. It would just lean on whichever pathway was left open.
So the solution wasn't a single hit. It had to be a combination, a 1-2 punch designed to take out both of the cancer's growth engines at the exact same time.
And right here, this is the smoking gun. This colorful square is a heat map that shows what happens when you combine the two drugs. Green means, meh, not much effect. But that bright red area, that's synergy. That's where the two drugs work together to become way more powerful than either one alone, completely shutting down the cancer's growth. The code was finally cracked.
Now, this is so much more than just a cool detective story that happened in a lab. Uncovering this backup plan, and especially the key players involved, has some really huge implications for treating actual patients.
And to prove this wasn't just some weird thing happening in a petri dish, the researchers looked at tumor samples from real TNBC patients. Just compare the normal tissue on the one side to the TNBC tumor next to it. All of that dark brown stain you see, that's our bodyguard protein, PRR5, and it is everywhere in the actual tumors. This confirmed their theory was absolutely relevant in the real world.
So, let's do a quick recap of what we've learned from this whole investigation. First, TNBC is incredibly clever and can outsmart single drug treatments. Second, it does this by using a backup growth pathway to survive. Third, the key to that backup plan is a bodyguard protein called PRR5. And finally, the best strategy seems to be a coordinated 1-2 punch that blocks both pathways at the same time.
And that brings us to our final and maybe most exciting question. The drug combo works, yeah? But what if we could be even more precise? What if we could take out the entire system just by disabling one crucial link in the chain? Could targeting this single bodyguard protein be the real key to disarming one of cancer's deadliest weapons? The investigation, thankfully, continues.